Welcome everybody. Welcome to this session on controlling and predicting microorganisms in food ecosystems. My name is Pau, and I'm going to co-chair this session together with Statis from University of the Aegean. In the session, we have 6 presentations, and we also have 80 posters, and I invite you to go and look at all these exciting posters after the session. We are ready to move on. So the first presentation will be given by Hanne Årslev Jensen from the Technical University of Denmark, and the title of that presentation is Effect of Dehatting and Storage in Modified Atmosphere on the Shelf Life of Shell-on Prawns. And those are cold water prawns, Pandalus borealis. So please, Hanne, the floor is yours.
Thank you, Pau. Thank you very much. Hello, and thank you for coming for my presentation. I'm an industrial PhD student working for Royal Greenland Seafood and the National Food Institute in Denmark, and will be presenting some of my work from my PhD. You, maybe? No. The cold water prawn Pandalus borealis is one of the most commercially important shellfish of the North Atlantic and the Arctic Ocean. It is a sought-after delicacy due to its sweet taste and bright red color. As a species, it is distributed over most of the northern hemisphere. And whole shell-on prawns are caught offshore by large trawlers, and after removal of bycatch and sorting by size, the products are cooked for 3 minutes, chilled and frozen, and packed for distribution. This occurs within 3 hours of catch to ensure the best product. Today, these products are sold as frozen, either in these retail cabinets where you can scoop up the amount of shrimps you want, or they are sold in smaller plastic bags. But industry has shown interest in marketing these products as a refreshed, chilled, ready-to-eat product, but very little information is known about the shelf life of these kind of products. Furthermore, industry has a large issue with miscolored heads, as seen by this picture, the arrows. Here we can see that our heads are darker colored compared to others. The cause for this is still unknown, but we do know that it's not a risk to human health. It just makes them look less appetizing. In the Scandinavian countries, you don't eat the heads, so it's not really a big problem. But we don't like looking at them. So the industry is actually sorting these heads on the trawlers. So we'd actually are thinking about what does it mean if we remove these heads. So we set up 3 research questions to be answered. First, we would like to investigate the effect of modified atmosphere storage on the sensory shelf life of these products. We'd also like to investigate how dehatting increases shelf life. Does it have an effect? And finally, we would like to investigate what microorganisms are responsible for the sensory spoilage occurring to these products. So to investigate this, we first had whole shrimp and dehatted shrimps, where we looked at the tail meat. We then packed it in either modified atmosphere or air and stored it for 5 degrees at 21 days. During this time period, we looked at sensory evaluations. We measured the total viable volatile nitrogen, and we also did aerobic plating. Furthermore, we did diversity studies based on 16S rRNA amplicon sequencing on fresh and spoiled samples to investigate how the diversity changed over time, and if we were able to see any differences between samples or storage methods. Furthermore, we also identified the spoilage microbiota by 16S rRNA Sanger sequencing of a representative amount of isolates from spoilage. And finally, we also wanted to actually see which kind of these isolates, or what isolates are responsible for the spoilage. So we looked into the spoilage potential and the spoilage activity by inoculating shrimps with high numbers of each isolate, and did sensory evaluation, aerobic plate counts, and total volatile nitrogen. So looking at the microbial growth, here we have the black lines are whole shrimp stored in air. The pink lines are tail meat stored in air. The green lines are whole shrimp stored in modified atmosphere. and the purple lines are tail meat stored in modified atmosphere. Based on this graph, we can see how modified atmosphere appears to slow down growth. And we also have an indication that actually the tail meat has a less growth or slower growth compared to whole shrimp. So looking at the sensory evaluations, we can see that whole shrimp stored in air has a sensory shelf life of 7 days, whereas tail meat in air had a sensory shelf life of 11 days. But modified atmosphere increased shelf life. So here we see for whole shrimp a shelf life of 18 days, and the tail meat had a shelf life for 21 days. So already now we have the answers to 2 of our 3 research questions. First of all, MAP does increase the sensory shelf life with more than 10 days, as we expected. And furthermore, deheading actually also increases shelf life with up to 3 days. So looking at the product changes occurring during storage, here we have the red lines being the oxygen content in the samples, and the blue lines are the CO2 content. These are the results for air-stored samples, and we can see that after approximately 11 days, the microbial growth has consumed all of the air present in the samples, which is quite exciting, I think. Maybe I should have used some other plastics, but it doesn't appear to have an effect on growth because the growth has already reached a high level. Looking at the modified atmosphere packaging in the gas composition, we don't really see any unexpected changes. We see an equilibrium after less than 4 days, but after this, the content appears to be consistent. So looking at the pH of the samples, here we have used the same colors as before, and we can see that initially the pH is around 7.5, and after 4 days we have reached a level between 8 and 8.5, but this remains consistent throughout storage, indicating that the microbiota does not affect pH. So then looking at the development of the total volatile nitrogen, here the same colors are used as before. And we can see that after 11 days of storage, air storage sample have begun production of total volatile nitrogen, and actually reaches quite high levels, not really seen in literature before. Whereas for modified atmosphere, this doesn't happen until day 18 and day 21. So this made us curious about who's actually responsible for this production of TVN. So we decided to look at the results from the diversity studies. Here we can see the beta diversity of the samples, and we can see that there is a difference between the fresh and the spoiled samples as we expected. The microbiology of the samples appears to be less diverse over time, which is expected because, yes. Furthermore, we can also see that there's a difference in the beta diversity between samples stored in air and modified atmosphere. And we also see that the effect of only looking at the tail meat also has an impact of the diversity. So from this, we looked at the 16S rRNA Sanger sequencing of the isolates. And we can see that in air, the dominating species is Pseudoaltemonas, whereas in modified atmosphere, it's Cyanobacterium. Only supplied by some other species. But we also see a difference in storage methods with the whole and the tail meat. So now that we know this, we would like to look into which of these bacteria are responsible for the spoilage. So we decided on choosing a representative amount of isolates, in this case 24 in total, for both storage conditions. So we looked at Pseudoaltemonas, Sucrobacter, Glutamicibacter, and Cyanobacterium from air stored samples. And we looked at Cyanobacterium, Suchomonas, Photobacterium, and Pseudoaltemonas from modified atmosphere samples. Then we did the sensory evaluation of the inoculated samples and found a sensory spoilage potential of 89% for Pseudoaltemonas in air, whereas Sucrobacter, Glutamicibacter, and Cyanobacterium did not appear to have any sensory spoilage potential. For modified atmosphere, Cyanobacterium had a sensory spoilage potential of 33%, whereas Suchomonas, Photobacterium, and Pseudoaltemonas in modified atmosphere had a spoilage potential of 100%. Based on this, we decided to investigate their ability to produce TVN. In this is a measure of how much TVN a single colony is able to produce, which is the spoilage activity. Here we can see that Pseudoaltemonas has a spoilage activity at minus 7.5, and as expected, none of the others has any spoilage activity. So this is likely the dominant or the cause for the sensory spoilage of the air stored samples. Looking at the samples for results for Carnobacterium in modified atmosphere, we have also a similar spoilage activity of -7.1. But Suchomonas photobacterium and Pseudoaltamonas has high activities, spoilage activities, but we also know that they are present in lower levels in the samples, indicating that it's probably a combination of these 4 species that are responsible for sensory spoilage in modified atmosphere. So the take-home message from my presentation is that MAP does increase sensory shelf life, and it's with more than 10 days. And deheading also increased shelf life with approximately 3 days. But industry has to decide if it's worth it. To remove these heads on board of the trawlers, which are already packed with a lot of equipment, it would mean that they had to add another process step, which is very hard on such small places or space. But they do have a lot of waste due to removal of these heads based on actually the catching area. So industry must decide, is it worth it to remove these? They might have less waste, which is a good topic these days, but it is adding a process step. Then we also found out who was responsible for the spoilage, and we found the spoilage microbiota does differ between storage methods, with Pseudoaltamonas being the main spoiler in air storage samples, whereas a combination of 4 bacteria was responsible for storage in modified atmosphere. So yes, this is the results of my work.
At this point, it is my job to read the questions that has been forwarded in the chat function of the system we are working in. And there are no questions that has been forwarded as far as I can see. So please correct me if you see something else from your side. Now, as there are no questions,
there is one question from here, Professor Dalgar. There is one question from the
please,
audience.
I don't see it. So please read the question.
Yes.
Good morning. Frank de Vliet from Kent University. Thank you for your presentation. It was really nice, very interesting. I was wondering, when you were testing the spoilage potential of your isolates, did you test them in air conditions always, or did you alter also doing this test in anaerobic or in MAP conditions?
Thank you for your question. I imitated the storage conditions when they were isolated, so the air stored samples were assessed in air, and the modified atmosphere stored sample was assessed in modified atmosphere.
Okay, thank you.
Another one question, yes.
Inge Dorte Hansen, Bio Veritas. As from a practical point of view, the microorganisms you found being responsible, would they show up on a generally recognized method for detecting total counts in these products?
Yes, they will.
Is there any other question? I have one question. Congratulations, first of all, for your interesting presentation. Probably the question is stupid, but I want to be sure. The Pseudoalderomonas is a facultative anaerobic bacterium, I suppose. Okay, thank you.
Thank you.
Thank you, Hannah. Then I think we will move on to the next presentation. The next presentation in this series is a little bit differently presented. I have been informed that this is a recorded presentation. The presentation is given by Micaela Alves Rubio, I think, and the title of the presentation is Modes of Action of Rosemary and Dibaromyces hanseini against Aspergillus westerdijkiae in dry cured meat matrix. So, next presentation, please.
I'm Micaela Álvarez from the Meat and Meat Products Research Institute of the University of Extremadura in Spain, and I'm going to talk about the work entitled Modes of Action of Rosemary and the Dibaromyces hanseini against Aspergillus westerdijkiae in dry cured meat matrix. This work has been carried out during a predoctoral stay in the Instituto Politécnico de Bragança in Portugal, and it has been completed in the University of Extremadura. I would like to begin talking about the microbial population throughout the processing of dry cured fermented sausages, with changes being the lactic active bacteria, the main microbial group, followed by the molds. The latter growth on the surface of the products during the ripening. These molds have some advantages as the promotion of homogeneous dehydration, color stability, and flavor development due to their proteolytic and lipolytic activities. On the contrary, other molds are able to produce spoilage, including texture and smell alteration, or even allergies and infections. From a food safety point of view, the main problem is the possible production of mycotoxins such as aflatoxins or ochratoxin A, OTA. The OTA is the most frequent mycotoxin found in dry-cured meat products, which are regarded among the most important contributors to the chronic dietary exposure to this mycotoxin, as it is suggested in the last scientific opinion of the European Food Safety Authority about the risk assessment of OTA in food. The OTA has a lot of toxic effects as nephrotoxicity, genotoxicity, or neurotoxicity, and this fungal secondary metabolite has been classified as a possible carcinogen. Although Penicillium nordicum has been described as the main OTA producer in dry-cured meat products, the species Aspergillus westerkii has been also considered a potential OTA producer in this kind of products. To avoid its presence, the meat industry commonly uses synthetic antifungals allowed by the European legislation, but some studies have been suggesting the potential toxicity of some of these compounds and the development of allergies in children. To avoid the use of synthetic antifungal, the new strategies are based on the use of natural agents. For example, the biocontrol agent rosemary and the yeast Variomyces hanseatia has successfully decreased the OTA produced by Penicillium nordicum in dry-cured fermented sausages. The rosemary is an herb frequently added during the processing of dry-cured fermented sausages in Europe, and the Variomyces hanseatia is the most frequent yeast isolated from these products. The efficacy of yeast by control agent derives from its mode of action, so the knowledge of the metabolic pathways involved will help to optimize the application method during processing and to make more effective combination due to possible synergistic effects. There are different techniques for evaluating the modes of action of control agents as the growth measure, the microscopic evaluation, or the use of molecular techniques as transcriptomics, proteomics, or metabolomics. The proteomic is a robust technique which has revealed that the relationship between a DNA sequence and protein structure is less predictable than previously thought, since the same sequence can give rise to different proteins due to post-translational modification during its intervention in biological processes. This technique can be used to understand complex metabolic pathways and the changes produced by the control agents like the rosemary and the Variomyces hanseatia through the increase or decrease of proteins by comparing the proteome of Aspergillus westerkii and different conditions through comparative proteomics. As the effect of these biocontrol agents against Aspergillus westerkii has not been studied till the moment, the main objectives of this study were to test the antifungal effect of rosemary leaves and rosemary essential oil and the Variomyces hanseatia on the OTA production by Aspergillus westerkii in a dry-cured fermented sausage model, and to elucidate their modes of action through the use of proteomics. For achieving these objectives, 3 different batches were performed in a culture medium made with lyophilized dry-cured fermented sausage salchichon. After plating the medium, a sterile collagen casing was placed on its surface to simulate the real product. A control batch without biocontrol agents, another with rosemary leaves on the agar and rosemary essential oil applied on the surface of the casings, and finally one with the Variomyces hanseatia added before plating the medium. The ochratoxigenic mold was plotted on the center of the plates. Then, they were incubated for 15 days at similar conditions of dry-cured fermented sausage processing. 3 days at 22 degrees and 90% of relative humidity, and 12 days at 15 degrees and 85% of relative humidity. After incubation period, samples of mycelium were collected for OTA production and the proteomic analysis. The OTA extracted by cultures was analyzed through an HPLC FLD, and the proteomic with a Q exactive plus. For proteomic analysis, the mycelium was lysed and the lysates were partially run in electrophoresis and in gel digested. The bands were subjected to a process of reduction alkylation, and then the bands were digested with sequencing grade trypsin and protease mass for 1 hour at 50 degrees. The results showed that the control batch had huge total concentration of more than 180,000 nanograms per grain, indicating that this mold is highly adapted to meat products. Despite the result, the biocontrol agents were able to reduce the OTA in a range between 74 and 88%. This result agree with previous studies about the effect of rosemary in Penicillium nordicum. In this case, the herb decreased the OTA concentration up to 62% in the same culture medium. Rosemary essential oil also decreased the OTA produced by other molds as Aspergillus carbonarius, Aspergillus ocraceus, or Aspergillus westerdicae. On the other hand, other strains of the Variomyces hanseatia have shown antifungal activity against Aspergillus westerdicae in commercial media, and the same strain used in the present study was able to decrease the OTA produced by Penicillium nordicum and Penicillium verrucosum in meat matrices. Proteomic analysis of the batch rosemary plus rosemary essential oil revealed a reduction of the abundance of the proteins from the PKS-ER domain, which is involved in the synthesis of a polyketide synthase. This protein is indispensable to form the ocratoxin beta and OTA precursor. Other proteins from the cell wall integrity pathway was also altered, as the DLC, which regulates the actin assembly during endocytosis and septopositioning, and the KIR9 related to the synthesis of cell surface polysaccharides. In addition, the cell wall protein FIA, involved in the survival response of toxigenic molds in the presence of toxic metabolites, was increased in abundance. Similarly, the yeast also affected to a protein from the PKS-ER domain, and also to another protein involved in the OTA biosynthesis, the non-ribosomal peptide synthetase, known as NRPase. This protein is indispensable for the union of phenylalanine to the ocratoxin beta to form the ocratoxin B. The cell wall integrity was also altered by different proteins involved in the actin binding and filament formation, and the actin and safety locations. The KIR9 protein was also diminished in abundance. Interestingly, one protein from the MAPK cascade was also reduced, suggesting a possible high sensitivity to cell wall stress agents. Summarizing, this figure represents the mold cell and the main targets of the biocontrol agent's action. The rosemary plus rosemary essential oil is represented with green arrows and the Variomyces hanseatia with orange arrows. The most important result is that both agents decrease the OTA concentration, affecting mainly to the OTA biosynthetic pathway and to the cell wall integrity by altering the actin, the cell surface polysaccharides, and, in the case of the yeast, the response to this damage. All these findings are collected in the final conclusions. Rosemary, together with its essential oil and the Variomyces hanseatia, causes a large decrease in OTA production by Aspergillus westerdicae. Their antifungal activity is likely related to their ability to reduce the abundance of several proteins involved in the OTA production and the cell wall integrity pathway. These biocontrol agents could be set within the hazard analysis and critical control points framework to minimize the hazard due to the presence of ocratoxigenic Aspergillus westerdicae in the acute fermented sausages. This work has been funded by the Foundation for Science and Technology of Portugal, the Spanish government, the government of the region of Extremadura in Spain, and the European Union. And finally, thank you for your attention.
Thank you very much for this detailed and very extensive study. There are no questions in the chat, so Statis, can you take over and evaluate if there are questions from the floor?
Yes, of course. Is there any question for Dr. Arvales? I have one question, Dr. Alvarez. Congratulations for this nice talk. I want to ask you. I want to ask you if you have tested only the rosemary leaves without the essential oil to see if you have any effect.
Well, I tested in this case against Aspergillus westerdickii. We don't have a good results because didn't change the outer production alone. Was only when the rosemary leaves were in combination with the essential oil, because there's a synergistic effect.
Okay, thank you. There is
Now
one question from the audience. Yes, please.
Congratulations for your exhibition. I just have a question. If you characterize
We don't
I can hear. No, I can hear.
I have a question. If you characterize this essential oil, can you hear me?
I can't hear well. Sorry.
Work? Okay.
Now, thank you.
Okay. So congratulations first for your work, and then I just ask you if you have the chemical composition of this essential oil, or if you tried a different rosemary essential oil in different tests, maybe.
Yeah, we did the essential oil part from our rosemary leaves with a Clevenger distillation. I know the composition of the essential oil, but this is part of other studies, so I can't say a lot of this. But in this case, there's a synergistic effect. Maybe it's not only the effect of the essential oils, also the effect of the part, the aqueous part, okay, from the rosemary. But it's similar to other essential oils from rosemary. So the only difference is that I did the essential oil, and then I use it in some weeks, not in too many times, like commercial essential oils. So it has more varieties of this kind.
Okay,
compounds.
thank you very much.
So
Thank you very much. Yeah, I think we can. I think we are right on time, and that we should move on. Or unless, do you have more questions from the floor?
Yes, there are no questions. We can continue.
Thank you very much. So now we go to the third presentation, and this time the presentation is going to be given by Jasper Bannenberg from University of Wageningen, and the title is Listeria monocytogenes Undetected. Inter and interspain variability influences the detection chance of enrichment-based procedures. So please, the floor is yours.
Okay, thank you for the introduction. So as mentioned, my presentation is about the detection of Listeria from food products. So I focused on the detection and how Listeria can remain undetected with the current procedures. So Listeria is most often found in ready-to-eat products like soft cheeses, cured meat, and smoked salmon. But there are some problems with the detection, such as the pathogen levels on food products can often be very low. And according to European law, you need to be able to show absence in 25 grams of ready-to-eat food products. Furthermore, the cells can often be damaged due to the food processing, and this can affect the physiological state of the cell, and which can in turn increase the lag duration during the enrichment. And this can even lead to false negative detection. So in short, the current gold standard is the ISO 11291 enrichment protocol. So you start with 25 grams of food product, and you do a primary enrichment of 24 hours in half Fraser broth. Then afterwards, secondary enrichment in full Fraser broth. After both enrichments, you plate out on a selective aloe agar. Then you have to plate onto non-selective plates to do confirmation steps. So as you can see, this protocol is quite long and quite labor intensive. But for my study, I was actually interested in this first primary enrichment, where you really have to focus on the resuscitation of an outgrowth of damaged cells. So we want to know more about what you need to optimally let these cells recover. So I started with a set of 23 food-related Listeria monocytogenes strains. So you wanted to have a reference condition to compare our results to. So the cells were just pre-cultured at 30 degrees, then serially diluted to enrich according to the ISO at a concentration of around 2 log CFU per ml. And then we wanted to have the effect of stress. So we chose 60 degrees heat stress. So all the cells were stressed for 1 log reduction according to their D value. Again, serially diluted and enriched. Then following the 24-hour enrichment, I did plate counts every 2 hours with biologically independent reproductions. I fit a microbial growth model and estimated growth parameters such as lag and growth rate. And also for some strains, I also looked at the effect of pH stress. So what we can see is that the growth rate was quite independent of the history. So it doesn't matter if there is heat stress or not. The growth rate was similar. So in this graph, first I want you to focus on the blue points. Those are the reference condition without any heat stress. So actually what you can see is that there is a lag duration. So after incubation, they have to get used to the antibiotics in the medium, but there's not really much difference in their lag duration. But when we look at the effect of heat stress, we can see that there is much more variation between strains. And we could actually identify some strains that are able to recover fast, a lot of them who have some average recovery, and some who really had a very slow recovery time. So then we wanted to see, okay, how can we explain this variability? So first we looked at the biological variability. So in blue is the reference, in gray after pH stress, and in red after heat stress. So the biological variability tells us something about the variation between reproductions. So as you can see, if we use heat stress, there is more variation between replicates due to the effect of the heat stress itself. But if we then look at the variability that is caused by differences in strain, we can see that there actually, for a reference condition, there's not really much variability. But if you look at the pH and even much more the heat stress, we can see that a lot of this variability is actually caused by differences at strain level. So for this, we used a scenario analysis of an enrichment when you start with 1 cell in 25 grams of product. We set a detection threshold of 2 log CFU that they have to reach in 24 hours, because after the primary enrichment, you take 100 microliter and add it to full Fraser broth, so you need to have at least these thresholds. And when we can see that for these fast recovering strain, they are actually able to reach these detection thresholds. But if we then look at the average recovering strains and the slow recovering strains, we can see that in these conditions, after this heat stress, we can see that they actually do not all reach this threshold. So there's a chance that they have a false negative result based on their differences between strains. So we can maybe already see that half Fraser broth might be too selective if you have really damaged cells, and it might be not sufficient for stress recovery. So now I want to focus, I talked about interstrain variability, now I want to talk about variability within a single strain. So we have a population of cells and a single cell, and the difference between this is that a population can exist of cells who are all in a very different physiological state. and they can actually display quite a bit of heterogeneity in how this physiological state is in the population. So now we wanted to look at what is the effect of single cell strains, because you need to be able to quantify very low concentrations with the enrichment. And for that we used flow cytometry. So in short, with flow cytometry allows you to target single drops with single cells in it, and you can analyze them based on their size and complexity or fluorescence. So I measured the single cell outgrowth by taking a fast, average, and slow recovering strain that I mentioned we got from the first experiments. We wanted to see the outgrowth in, of course, the enrichment medium, but also BHI as a non-selective control. Then 384 Wells plates were filled with a single cell in each well with a cell sorter. Then we measured optical density at 30 degrees, and then for each well we could actually calculate the time it takes time to reach a certain increase in optical density. So if you look at the reference condition again, without any heat, without any stress treatment, on the top we can see in BHI that actually the outgrowth is very homogeneous. There is not much variation between these single cell events. If we then go to the bottom at half Fraser broth, we can already see that, of course, the lag duration is longer, as we've seen before. And there's also starts to become a little bit more heterogeneity and a little bit more separation between the different strains. And this is really becomes much bigger if you look at heat stress. So you can see that in non-selective medium, in BHI, that the slow recovering strain really starts to have a lot of heterogeneity, that some single cells really grow out much later than average. And if you look then at, again, in the enrichment broth, we can see that, the outgrowth at a single cell level can be really heterogeneous, and that can even lead to some cells really growing out much later than others. So now we again wanted to quantify, okay, can this single cell heterogeneity also impact the detection? So first, on panel A, we simulated the growth in a well. So this is the growth in a well, so it needs around 24 hours before you see the optical density threshold. So we can set the time of detection that you would expect if there's no lag in the well. And then we also estimated when you want to detect one cell in an actual enrichment, there is a maximum lag duration where you can still reach the threshold that I mentioned also before. So combining these, we can actually estimate that after a certain time to a detection, a certain amount, when you reach this optical density threshold, that really, after that, the cells are really at a chance to be a false negative result and can impact the detection. So here again, I showed the outgrowth in enrichment broth for the fast, average, and slow recovering strain, and in the red boxes are now these false negative chances. So we can actually see that for the fast and average recovering strain, as seen before in the simulations, there's not really a problem if you have just reference cells, but already for the slow recovering strain, they're actually around 27% are already have such long lag duration that there's a chance that you will miss them during detection. And we look at the effect of heat stress. We can see that for the average recovering strains in these conditions, more than 50% is in this area for a false negative result. And for this really slow recovering strains, as we've seen before, there really is a very big chance that the outgrowth of these single cells is just too long, that there's a chance that you will miss them during detection. So some take-home messages. I've shown that strain variation can impact detection probability. There's the difference in outgrowth in enrichment broth of different strains can really can really differ a lot if you have a stress history. But even within a single strain, there's also a lot of heterogeneity at the single cell level. And this heterogeneity is important to take into account when you only have a few cells on a food product that you need to enrich. So this actually has direct implications on the detection of Listeria monocytogenes using the current ISO protocol. And as a last point, enrichment in half razor broth might actually be already too selective to efficiently repair damaged cells in this 24 hours of primary enrichment. So thank you for your attention. I want to thank my promoters, and here I have mentioned the papers that where I in more detail describe this this data. So thank you for your attention.
Thank you very much. And we will move to questions. There are no questions in the chat. If there are no questions from the floor, I definitely have a questions, but I think we should go to the audience first and hear if there are questions to this presentation.
Yes, please.
Good morning. Thank you for your presentation. I have a couple of questions. My first question has to do, I was slightly confused with those two figures you had, making a distinction between strain variability and biological variability. I mean, to my perspective, strain variability is a biological variability. So did you most likely mean experimental variability in one of your figures?
Yeah, so for the biological variability, I looked at the difference between reproductions and the strain variability.
Experimental replicates, you mean something like
yeah,
that?
Yeah, within the replicates and the strain variability.
So maybe, I don't know, but to my point of view, and I think it's also, widely reported as such in the literature, this is not actually a biological variability. Maybe it should be, reported as experimental variability, uncertainty, or something like that, because strain variability is maybe the most important source of biological variability. So having this distinction is slightly confusing, just for,
Yeah.
And my second question is, why did you prefer to actually use the 1D value criterion for deciding for how long the heat stress would be applied, which most likely means that you had different times for each strain,
Yes.
and not actually apply the same duration of heat stress, so that you could also take into account maybe variation among strains that has to do with other parts of the inactivation curve, not just the linear part, taking into account the shoulder.
Yes. Yeah, so thank you for your question. So at least we see that for Listeria monocytogenes strains, there is very large variation in D values. So I think we chose to use a similar level of reduction so that we could have as much level playing field as possible, so that we can really see, okay, what is the effect on the individual delect durations. There are multiple ways in how you can normalize this, but we chose to have a similar level of reduction for all the strains because there's such a big variation in heat stress reduction.
Okay, thank you very much.
Thank you. Should there be one more question? That is not the case.
No, there is question.
There is a question.
Yes, thank you for an
Let's take
excellent.
one.
Thank you for excellent talk. Do you think this is the reason why we see that certain sequence types, like 1-to-1, is frequently isolated from a factory environment, that we don't really see the outbreak strains isolated from factory environments?
Yeah, so I definitely think there is a bias that some strains or sequence types are more likely to be detected, but we did not really see a correlation, at least in our subset, with respect of their lag duration and their sequence type or origin. So I think that there is a bias in certain strains that are more easily to be detected, but I don't have any conclusive evidence that this also relates to sequence types specifically.
Thank you very much. I think we have to stop here because we are actually on time. Huh? I'm very sorry.
I'll see you later.
Yeah. We have been instructed to stay very specific on time, so I think we should do that. At this point, I will give the word to my co-chair, Stathis, and he will take over from here. Thank you very much from my side.
Okay, thank you. So the next presenter is my co-chair, Professor Dalgaard from the National Food Institute of Technical University of Denmark, and he's going to speak up to speak us for Clostridium sporogenes, a surrogate for proteolytic Clostridium botulinum, development and validation of extensive predictive model using data for growth and time to toxin formation. Professor, the floor is yours.
Thank you very much. As this conference takes place in Greece, in Athens, I would just mention to you that all my co-authors of this study, they're all in Denmark at the time of the study, and they're all Greek. So I just thought I would bring that up. And so I'm going to talk about Clostridium botulinum. I'm going to talk about surrogates, and I'm going to talk about botulism. I mean, botulism is a very serious matter. A large number of people with botulism die. Fortunately, it's very rare. So we are not completely out of control. It's a rare event, fortunately. It's caused by both cyclotolerant and mesophilic variants of Clostridium botulinum. We call them group 1 and group 2. As you can see to the right here, these are from a phylogenetic point of view, they're completely different, and there definitely should not be the same species. It has also been suggested that group 1 should be called parabotulinum. Now, Clostridium botulinum group 1 is very closely related to Clostridium sporogenes, as you can see to the right. And today we are going to use Clostridium sporogenes as a surrogate to develop a model for Clostridium botulinum group 1. I should say that these two species, I mean, both of them can actually produce botulinum neurotoxin, but they also both have variants that do not produce that toxin. So we have selected strains that do not produce the toxin, and we have used those for development of a model. So management, we clearly have to be very efficient in managing this hazard. We have no regulation to guide us on specifically what to do. Fortunately, we do have guidelines, and the guidelines I think are very well known to pretty much all of us. Just keep the temperature low. There will be no problem with group 1 Clostridium botulinum. Have low pH if we do not have temperatures below 10, or high salt in water phase above 10%, or we use the botulinum cook. That's all fine. And this is probably the reason that we have a low probability of finding botulism or seeing botulism. But there is, of course, a huge problem here, and that is we have a large number of products where it is really not feasible to have pH below 4.6 or up to 10% salt in the water phase. Now, when those products are developed with medium or low levels of salt, and there is increasing focus on these products with low salt, we can use challenge tests to document safety of the recipes, that we find a combination of conditions that is actually managing growth of Clostridium. The problem here is that the challenge tests are expensive because it's an organism that can be really, really, really dangerous, and there are very strict conditions on how laboratories should be organized, and they're also quite slow. So we need something new. We need a tool that can assist us in this product development for safe recipes controlling Clostridium botulinum. And that is exactly the objective of this study. And the approach we have taken, I've already mentioned. We are using a mixture of non-toxigenic Clostridium sporogenes as a surrogate for Clostridium botulinum group 1, and we use data from the literature, both growth and time to toxin data for Clostridium botulinum for evaluation of the model that we are going to develop. So the approach is that we generate data in the laboratory in broths, and we have a substantial amount of data, more than 600 growth rates. We then validate or calibrate our model to growth in processed cheese. That was That was a focus product in this study. And then we move on and we have more data in well-characterized processed cheese for evaluation of the model. And then we collect all the information we can find in the literature, more than 100 growth rates for a large number of toxin-producing Clostridium botulinum strains, and also a very large amount of time to toxin data, and we use those for evaluation of the new extensive model. So the data you see here, the growth rates of Clostridium sporogenus, and how we have developed a very classical cardinal parameter model for the effect of temperature, pH, and water activity. And the water activity is due to changing concentrations of sodium chloride. Very standard. We included in our model also the effect of organic acids. They are always present in processed cheese, where we find primarily lactic, citric, and acetic acid. But we also want to use the model more broadly, so we included benzoic and sorbic acid as well. And they are listed here in the sequence of their sensitivity. So Clostridium sporogenus was most sensitive to citric acid when we measure this as undissociated concentration of acid, and least sensitive to acetic acid. Now, as processed cheese was an important product in the study, we included the effect of phosphate melting salts. And you see here that we have orthophosphate, we have diphosphate and triphosphate, and the inhibitory effect of these melting salts, I mean, they are in the product due to the texture and the properties of the cheese, but they have a inhibiting effect on Clostridium. And that effect is very different for those three, so that we need to take that into account. We cannot just say the total amount of melting salts, because that would not be precise. We then combine all these terms that we have developed into one big model. You see that within the red box here. I don't have time to go into the specific details. I want to mention the last small term, the interaction term, because it is really, really important to take into account interaction between all the factors if you want to predict the growth boundary. And that is what we want here. We want to predict no growth conditions. So we have done that, and we have used the classical Lamarck approach. Now, just a few characteristics of our study. We have to take into account the effect of lipid in the product on the concentration of benzoic and sorbic acid in the water phase of the product. So that's really mass balance modeling. We also had to develop a specific, that was a qPCR method for enumeration of Clostridium in food that include other microorganisms, so real food. And then we have to do a detailed characterization of all the products that we study in challenge test, including actually a new method for quantification of the specific melting salts. Finally, we use the classical approach of bias and accuracy factors, and also the percentage of failed dangerous predictions to evaluate our growth and time to toxin formation models. Now, we developed this extensive model with 11 factors included, and we compare that model to available model for Clostridium sporogenus and for Clostridium botulinum. First, to our new model. What we can see is our data that was mainly for processed cheese. The model predict growth to be slightly too fast, 17%. And we have similar performance for literature data. And the literature data was primarily from meat, and here the model performed very well. So the model is acceptable. If we go to the data for models that are less extensive, include only a few factors, then you can see they cannot predict growth in cheese products. And the reason is very simple. They do not take into account the effect of organic acids. Therefore, the predictions are too fast. And then they predict slightly too slow in other products. And we have a same pattern for models for proteolytic Clostridium botulinum. There's also one model given failed dangerous predictions, and we cannot go into the details of that here. But that is, of course, really, really important that we don't see that for models. Now, if we move to the time to toxin models, and that is really important, important in the sense that There is a lot of data for time to toxin in the literature. There are very little data for growth of Clostridium botulinum. So we use these time to toxin data not because we want to predict time to toxin, because what we want to predict is conditions that prevent growth. But we want these data to evaluate our model. And we can see here that our model is transformed from a growth model into a time to toxin model by predicting the time for 3.3% log increase. This is the same approach used previously by Testake and Coopers quite a while ago. And you can see when we evaluate the model, it performs quite well. We have 2 failed dangerous predictions out of 237, which is a better performance than available models. And also our new model have a much wider range of applicability because it's more extensive. So we were quite pleased with these data. One thing that I would like to highlight is that when we have a complex model like this one, we can use it for product development. And this is really, now we're really back to the problem. When we take into account the effect of microbial interaction, then that allows us to develop or identify conditions that are where concentrations of individual compounds are below their MIC value. So they should result in growth, but due to interaction with the other factors, there are no growth. And this is really the Lemarck approach that allows us to do that. And our model here allow us to identify conditions for processed cheese at room temperature with less than 1% salt and with pH 5.8, where we have no growth and we even have a good safety margin to the growth boundary. Now, this brings me to the conclusions. We have a new model with a wide range of applicability. Please observe that I also say that this model can be used for aerobic conditions. We saw a number of cases where products stored in air allowed growth of Clostridium sporogenous. We have predictions with a good margin to the growth boundary. The model can be used to identify recipes that prevent growth and toxin formation. That can be done by businesses. I think authorities and advisory committees could also look at this model when they make guidelines. Guidelines, in my opinion, today are very unflexible. We need more studies, of course, to further increase the model, but it's already have a wider range of applicability than other available models. And when we have a complex model like this, it is very beneficial to include it in software so that the use of the models is facilitated, and also we can place around boundaries on the use of the model so that it is not being misused, which is in fact a challenge. Study was funded by the Danish Dairy Research Foundation, and the growth part of this study has been published. The toxin part is on the way. Now, at this point, I thank you for your attention, and I guess we can take questions if there are any.
So thank Professor for your talk. We have some questions.
Hi, this is Marion Wells-Bennick from NISO. Hi, thanks very much for your interesting presentation. I have a question related to the time to toxin, which is based on that 3 log increase. How do you deal with a very local growth? Let's say there's one colony growing in a larger batch, because we're not looking at liquid foods here. Processed cheese is bacteria cannot just swarm freely. How would you deal with that in your model? Let's say there is one colony growing in a kilo of your product, reaching that 3 log increase, but locally you may have neurotoxin produced. How would you deal with that in your model, for instance?
We will not deal with that at all, because we don't predict time to toxin. That is only for validation to show that our model has a reasonable performance. This model can only be used to predict conditions that do not allow growth and do not allow toxin formation. And that is done with a large margin to the growth boundary, a psi value of 2. Okay. This is really important, because otherwise people would start to use this model for risk assessment, and that would be a severe misuse of the model. We are really highlighting this in the paper, that you cannot use this model in that way. And that's also why I mentioned that when we include this in our software, there will be boundaries so that you cannot make that kind of prediction for time to toxin, because that's really not what the model is developed to do.
Okay, great. Can I have
This is really for those people who formulate safe recipes. It's not for those risk assessment studies.
Okay, great. And perhaps one other question, if okay. A very technical question relating to how you assess the counts of Clostridium sporogenes in the processed cheese. Did you do that via selective media or non-selective?
No, qPCR method that
Oh,
we
only
developed.
qPCR.
That we developed as part of this study, yeah.
Yeah, okay. Well, just to add to that, because the experience we have with particularly sporogenes, if you plate it on the typical media, the ones that turn black, the iron sulfide is very critical. If it's too high, like the standard media, and this is now incorporated in the new ISO method, you have to make sure that you use not too high concentrations of iron sulfide, otherwise you won't find them. But
Plate counting media for Clostridium botulinum in general is a nightmare.
Yeah. Okay. Thank you very much.
Thank you.
Thank you, Professor.
Time is...
Thank you for your interesting presentation. This is Ioannis Giavasis from the University of Thessaly in Greece. Just a general question. Do you know if the use of such modules using Clostridium sporogenes are accepted as, valuable, not valuable, but are they accepted from authorities from the European Union countries for challenge tests that can show that there is no growth of Clostridium botulinum? I mean, apart from the research part, would the company be able to use such a model and verify that its product is free of Clostridium? And I'm asking this because I once had to make a challenge study, well, a company wanted to make a challenge study for using Clostridium botulinum, and I found it almost impossible to find a lab in Europe that is entitled to do such tests. So would, apart from the scientific part and in the interest of all this, is this officially acceptable as a challenge test that could be sent, for example, to EFSA or to a European authority? And would that be able, such a model would that be able to also cover the psychophilic strains, or is this completely different story? Because many products suffer from actually these psychophilic strains during refrigeration. Thank you.
Well, thank you very much. This is a very relevant question. I will try to answer shortly because otherwise we will really run over time. I will take the last question first, the psychotolerant strains. We already developed a model for those. It was published last year. So that model is available. Now we come to how should we use these models? Now for Listeria monocytogenes, we developed similar models 15 years ago, and it took about 5 years and more for the Food Safety Authority to accept these models and say you can use them to make predictions. The models for Clostridium do not have the same precision as the models for Listeria monocytogenes. So our recommendation here is that you use the models to identify recipes that give you no growth, and when you have done that, then you do perform your challenge testing. So the model will allow you to make your challenge testing more specific and reduce the cost. It will not completely prevent challenge testing. The models are not at that stage yet, and I doubt they will get there. We have problems predicting lag times precisely.
Thank you.
Thank you.
So we should move on. The
Yes.
next presenter is Mr. Imed Dorbani from the French National Research Institute for Agricultural Food and the Environment. He's going to give us a talk on comparing effects of pulse light and UVC radiations on bacterial and fungal spores.
Thank you. So, good morning, everyone. My name is Imed Dorbani. I'm a PhD student in a mixed research unit at Avignon University and INRAE. My work and research are supported by Claranor company, and I'm so honored today to present you a part of my work, which entitled Comparing Effect of Pulsed Light and UVC radiations on Bacterial and Fungal Spores. So, as you probably know, microbial contaminations remains a major problem in food and even non-food industries, so as they produce undesirable effect on health and economy of public. So to ensure the safety and stability of and the quality of food products, different kind of decontamination technologies are available in the market based on thermal and non-thermal methods. And as one of a novel non-thermal decontamination technology, here is pulsed light. So here you have an example of a pulsed light machine implemented in high throughput chain production for metal cane decontaminations. So with this technology, we can achieve 3 to 5 log rejection on highly resistant microorganisms, including bacterial and fungal spores. And Claranor company is one of the leaders providing pulsed light machine for packaging, sterilization, even surface and clear liquid decontamination also. So the question is, what's pulsed light? So pulsed light is based on the release of high electrical tension on xenon flash lamps. You can see here in your left the schema. So the release of this high electrical tension generates a white light flash. This white light flash is characterized by a high intensity because we put a high power. And also, it's a fast processing because one flash occurs in approximately 250 microseconds and emitting in a broad electromagnetic spectrum covering UV, different range of UV C, B, and A, visible and near-infrared wavelengths. So here you can see the electromagnetic spectrum of pulsed light in your right, which called also polychromatic spectrum because it's emit in epochs in different wavelengths, and also compare it to the UV light based on low mercury vapor lamps, low pressure mercury vapor lamps. It emits a monochromatic spectrum at one peak at approximately 254 nanometers. So the objective of our study is to compare the resistance of bacterial and fungal spores to pulsed light and to UVC. For this, we selected strains, relevant strains. Here you have an example of the strain used in this study. We have strains used in biovalidation of industrial sterilization systems. You have also some strains belonging to Bacillus cereus group, relevant in food industries. And you have also particular strains, Bacillus pemilus, which were spores described as highly resistant to UV and cosmic radiations also. So here you have an example of the survival curves of two Bacillus pemilus strain as function of the fluence. Fluence is the dose of pulsed light and UVC. It's in joule per square centimeter. And as you can see, we achieve more 5 to 6 log rejection using the two technologies. So the two technologies are pretty efficient, inducing inactivation of Bacillus pemilus spores, as you can see here. The major difference between the two technologies is the time exposure, because for UVC, we need approximately 140 seconds to inactivate Bacillus pemilus spores, we need just 1 milliseconds to have the same inactivation level between the two technology. So pulsed light is a time-saving technology also. So using survival curves, we can calculating parameters, different parameters, like F2, it's the affluence for 2 log rejection, and F3 also for 3 log rejection, etc. So we did the same thing with other strains, and we obtained these results. So F2 in your left, and F3 in your right. So as you can see, the result demonstrated that there is the different strain expressed a different level of sensitivity to pulsed light and to UVC. And we also demonstrated that there is a relevant correlation between sensitivity to pulsed light and UVC for Bacillus spores. Sorry, and this result demonstrated also that there is that the UVC resistance is a good predictor for pulsed light resistance for Bacillus spores. So the inactivation mechanism of UVC is well established because it occurs through DNA damage, which is called also photochemical effect on spores and cells. But for pulsed light, it's not clear because some authors report photochemical effect because of the UV content of pulsed light, but there is also other effects on the spore structures. So we wanted to compare the effect of pulsed light and the UVC on Bacillus spore proteome. So for this, we used a proteomic analysis, a proteomic approach on Bacillus pumilus spores, inoculating on polystyrene carrier, and we treated this Bacillus pumilus spores with dose allowing 5 log rejection and 2 times the dose allowing 5 log rejection. The protein were extracted, and a shotgun proteomic analysis have been performed. So proteomic analysis revealed that there is more impact on proteins when we analyze, we compare the protein abundance between untreated and treated samples, and the results revealed that there is pulsed light impact. Pulsed light impacted more spore proteins than UVC did. And also the result demonstrated that there is a dose effect for both processing, including pulsed light and UVC. So when we compare the two protein data set between pulsed light and UVC, the impacted protein, we observe that there is more, most proteins are specifically targeted by pulsed light than UVC. We performed also a scanning electron microscopy on Bacillus pumilus spores, treated and untreated. So as you can see in your right, this is treated spores of Bacillus pumilus with pulsed light, and we, as you can see, we observe some cell debris, but there is not a major degradation or major impact on the spore structures. So now what about fungal spores? So for fungal spores, we tested three different species, 2 Aspergillus and 1 Penicillium. And as you can see, compared to UVC survival curves, pulsed light is pretty efficient inducing inactivation of fungal spores, especially for Aspergillus brasiliensis and Aspergillus carbonarius, because with one flash at 0.75 joule per square centimeter, we can achieve approximately 6 log rejection on the tree mold. So when we add the results obtained in the same condition with the Bacillus pumilus spores and Geobacillus stereothermophilus, you can see that the difference between the resistance of bacterial and fungal spores is greater for UV than pulsed light. So this results demonstrated that the relative resistance between fungal spores and bacterial spores are pretty similar for pulsed light than UVC. We performed also scanning electron microscopy on Aspergillus brasiliensis spores, and this is a representative images of what I have seen in the scanning electron microscopy. And as you can see in your left, comparing to, in your right, comparing to the Aspergillus brasiliensis spore untreated Bacillus aspergillus spore, here you can see that there is several damages on the spore structures. Here you can see there is holes and big holes, and also releasing of some intracellular components in the extracellular environment. So to conclude, for bacterial spore, Bacillus spore resistance to UVC is a good predictor of pulsed light resistance. Pulsed light induces more spore proteins degradation than UVC did. And as we have seen, no structural damages induced on pulsed light treated Bacillus pumilus spores. And for fungal spores, pulsed light is more efficient than UVC for Aspergillus spores in activation. And also, pulsed light induces major structural damage on Aspergillus brasiliensis, as we have seen previously on scanning electron microscopy images. So to say last thing, pulsed light is efficient on both bacterial and fungal spores, but probably with different mechanisms. I would like to thank all my PhD supervisor, Frédéric Carlin, Catherine Duport, and Clara Nor Company supervisor also, and all the collaborators. I would like also to thank the chairs and also the committee organization of Food Micro. Thank you.
So thank you for this nice talk. Is there any question? Yes, please.
Yeah, so maybe this is something I didn't understand. Is it the light itself that disrupts the spores, or is that just a byproduct? And if it's the light, how exactly does it work?
On Aspergillus spores, there is a major destruction, as you have seen in the scanning electron microscopy. It's just the effect of the light on this, but not on bacterial spores, because the light have it's a powerful flash with high intensity and a broad spectrum. So it's the effect of the light.
Okay. So it's just so powerful it destroys.
Yes.
Okay. Thank you.
Thank you.
Yes, please.
Well, first of all, thank you for this very fascinating talk. It was a very good talk. Can you tell us a little bit more about the proteomic approach that you have used? Because you see that some proteins are degraded. How do you see degradation of proteins? Do you have peptides that disappear of the spectrum, or?
For proteomic analysis, we didn't see the degradation. We didn't investigate the degradation, but we compared the abundance of proteins between treated and untreated samples, and we apply filters with the statistical analysis. But the the future investigation will be on the exactly degradation of the proteins and the effect. Maybe there is oxidative effect on some proteins. So yes, it's.
Yes, please.
We know that UVC is quite relies on that it is a clear product and very thin layers. One would think that the same is valid for the pulse light. What was the size or the dimensions of the tool that you were using?
The tests were performed on water and also on a small surface of water, and also on surfaces like polystyrene and glass. But like UVC, pulsed light is low penetrating on other matrix, so it remains a surfacic treatment.
The next presenter isn't here yet.
I think there is a question.
Yes, that's why we have some time for some questions. Yes.
Hello, thank you for the presentation.
Thank you.
I saw bottles going through a machine and then the pulsed light.
Yes.
So now you're saying that you measured on a flat surface. Does it work, and do you see the same comparison with bottles, for instance?
I didn't perform tests on bottles, even no on cans, but on standard surfaces, just plain surfaces for the test. So I don't know if there is. It's quite efficient on packaging sterilization application, but I didn't perform a test on this.
Okay, and the strength of the light is. How did you compare that? You can have different hertz or strongness of the light.
Wavelengths?
Yeah. No, not wavelength. The strength.
I didn't did this.
Okay.
Thank you. Thank you for the question.
So, is there any other question? I have one quick question.
Yes.
Is there any advantage of this technique regarding the safety of the personnel who uses it? Is it a safe technique?
You have to protect yourself when you use this technology because there is a UV and high intensity, so we wear glasses and gloves.
Okay, thank you.
Thank you.
So I do not know if the next presenter is here or he is going to be connected online. No? No. So this session is finished. I want to thank all the presenters for giving their talks, and I want to thank all of you for being here. Have a nice break.